Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Advancing Atherosclerotic Cardiovascular Disease Trials Through Innovative Imaging Endpoints. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Medpace and Caristo Diagnostics who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas, including cardiology, oncology, metabolic disease, neuroscience, and antiviral and anti-infective. And next, I'd like to present Caristo Diagnostic, which is a global leader in the AI-powered heart disease detection, a spin-out from the University of Oxford. Caristo's award-winning technology is transforming cardiovascular care by identifying hidden coronary inflammation on cardiac CT scans. Backed by leading hospitals and scientists, Caristo is on a mission to save lives through early detection of cardiovascular and personalized prevention. Caristo has been supporting multiple pharmaceutical and biotech companies' development efforts for both preclinical and clinical stage drug trials. Next, now is my pleasure to welcome our speakers to today's event, and I'd like to introduce you to Dr. Rina Arriga. She is a board-certified cardiologist and physician scientist with over 15 years of academic and commercial clinical research experience. She trained in London and Oxford and obtained her PhD at the University of Oxford, where she held both clinical and academic appointments. At Medpace, she provides hands-on medical leadership to sponsors and internal study teams engaged in cardiovascular clinical research. Next, it's my pleasure to introduce you to Dr. Haris Antonides. He holds a BHF Chair of Cardiovascular Medicine at the University of Oxford and practices as a consultant cardiologist at the Oxford University Hospitals. He is the Director of Acute Multidisciplinary Imaging and Interventional Center, University of Oxford, and the Deputy Head of the Division of Cardiovascular Medicine. He serves as the chair of the British Atherosclerosis Society and is involved in several leadership roles within the ESC. He is also founder and chief scientific officer of Caristo Diagnostics, a University of Oxford spin-off company that I presented earlier. Now it's my pleasure to welcome Dr. Andrea Flannery. She is a Director of Clinical Trial Management at Medpace with a PhD specialized in microbiology and device-related infections from the National University of Ireland, Galway. Her therapeutic experience specializes in global cardiovascular clinical trials, including ASCVD, and leads the cardiovascular outcome trials group within Medpace. Now, with the introductions out of the way, it's now my pleasure to pass the mic over to Rina. So, Rina, when you're ready, you may begin.
Thanks, Sonia. Hi everyone, welcome to our webinar on imaging endpoints in atherosclerotic cardiovascular disease trials. Imaging is no longer a nice-to-have add-on in cardiovascular trials. It's becoming the foundation for smart and efficient trials. It's not just about tracking plaque burden anymore. Imaging is redefining how we design and run trials. Is giving us earlier and richer insights into the impact of treatment. But how do we image coronary inflammation, the root cause of atherosclerosis? One such emerging marker is the Fat Attenuation Index, or FAI, derived from coronary CT angiography. We will discuss this innovative FAI technology and its clinical relevance. We'll cover the considerations when designing trials with imaging endpoints, and then from theory to practice, how to operationalize these imaging endpoints in trials. It's my great honor to hand over to our guest speaker, Professor Haris Antonides, who has led the pioneering work in Fat Attenuation Index. I've had the privilege of working with Haris as a colleague when I was previously an academic cardiologist at Oxford. I'm looking forward to continuing our collaboration now in the context of advancing drug development. You're so very welcome, Haris. Over to you.
Thank you, Rina, for your introduction. It's a very great pleasure for me to be with you today to discuss about how to measure coronary inflammation using CT and particularly the Fat Attenuation Index technology. These are my disclosures. So in preventive cardiovascular medicine, we have been talking for a very long time now about personalized treatments, lifelong risk prediction, but in reality, the fundamental question is whether we want to go down the route of the one-size fits it all, the poly-pill approach, where you put a little bit of everything in a pill, you give it to everybody, and you don't have to care about personalized treatments, or going down the route, the more expensive route of precision medicine, where you need to invest heavily in identifying the right patients for the right treatments. This is where all the guidelines are heading to now. However, we still rely on simple risk factors based calculators for our cardiovascular risk. If we want to truly implement precision medicine, we have to start thinking about genetic scores, plasma biomarkers, imaging biomarkers to help us identify the right patient for the right drug. Looking now into the disease, the cardiovascular disease trajectory, we know that 6.5% of the population will develop obstructive coronary artery disease at some point in their life, and these will come with symptoms, chest pain, angina. However, we know that nearly half of the heart attacks, 50% of the heart attacks, happen among those who never had symptoms before, and they do not have obstructive disease. This is why now we focus more on the early identification, early detection of cardiovascular disease, because that's a window of opportunity to intervene and prevent heart attacks from happening. What do we need to have in our toolkit in order to start talking about early identification of patients at risk? We need to be able to detect the plaques, find the small non-obstructive plaques. We need to be able to characterize the plaques, tell whether they're calcified or stable or unstable, and more importantly, be able to find the inflamed arteries, because it's the inflamed arteries that give the heart attacks, and it's the inflamed plaques that rupture give the heart attacks eventually. And this inflammation precedes atherosclerotic plaque formation. Coronary CT angiography is probably the golden bullet that can give us all of the above, all this information. Indeed, is now the first-line investigation for patients with chest pain, and in a recent study in 40,000 patients, the orphan study, that were followed up for 3 years after they had a coronary CT angiogram, we have seen that only 18% of these people had obstructive disease, but 2/3 of the heart attacks happen among those without obstructive coronary artery disease, which means that if we focus on those with obstruction, we miss most of the cases that will give the heart attacks eventually. Inflammation is probably the most hot therapeutic target that is being worked by the pharmaceutical industry today. We know from the Lotoco-2, the Colcott, the Cantos trials that if we target inflammation, we reduce cardiovascular events. And the FDA has recognized this by giving the label to colchicine for cardiovascular risk reduction since 2023. However, the big question is, how do we find the people that we need to treat, the people who have inflamed coronary arteries? If we use plasma biomarkers, then these are reflecting systemic inflammation, not local coronary inflammation. In my group now in Oxford, in the University of Oxford, we have focused for many years on trying to understand how the arteries communicate with the surrounding tissue, with the perivascular adipose tissue. And we have seen that if you have an inflamed artery that secretes inflammatory molecules in the perivascular space, the fat cells in the perivascular space sense the signals and they get small and fat-free, as you can see in this cartoon. This happens before you see any plaque formation. Now, as time goes by, these changes in the perivascular space become more and more prominent. And when you treat the patients with an anti-inflammatory agent, these changes in the perivascular space recover. So they dynamically change in the response to inflammatory signals coming from the vessel. Depending on the type of inflammation now you have, you may get these very complex changes in the structure of perivascular fat with fibrosis, angiogenesis, and so on. Now, in the lab, if I'm able to take a biopsy of the perivascular fat, I can tell not only if the artery is inflamed, but I can tell what type of inflammation the artery has. However, getting biopsies for perivascular fat is impossible because you cannot open the chest. And that's why you develop a method that uses routine coronary CT angiograms and performs what we call virtual biopsy of the perivascular space to tell us about inflammation in the vessels. As you can see here, the coronary CT angiography gives us these black and white images. Perivascular fat is not visible. We developed this method that is called fat attenuation indexing that brings this information to the surface and turns these black and white images into colorful images. If you have a non-inflamed artery, it appears as yellow. If you have an inflamed artery, then you start seeing this red color around the artery. And if you have these complex changes, depending on the type of inflammation, you may have these patches of blue, which reflect more edema and more fibrosis in the perivascular space. In Oxford, we are lucky to have this unique infrastructure. We have this cath lab that is integrated into the same space with the photon counting CT scanner. This is a research cath lab, and a research scanner is dedicated for running trials, running primary research for us, for the university. And we use this infrastructure to validate these kind of technologies against intravascular imaging. And you can see here how this patient with an acute coronary syndrome has this highly inflamed plaque in this area that lights up as red in the perivascular fat analysis. When we developed this technology a few years ago, we realized early on that the basic technical metric that is called pericoronary adipose tissue attenuation was very variable, and it was dependent on technical parameters from the scanners. It was working only on the right coronary artery because of the more complex anatomy of the other coronary arteries. That's why we moved very fast into development of the fat attenuation index, which is the technically corrected metric of perivascular attenuation, and more recently to the fat attenuation index score that takes into account the anatomical, biological, and other technical settings, scanner settings that need to be taken into account to generate a reproducible tool that is applicable clinically. And it's this fat attenuation index score that has received the regulatory clearance with the label of biomarker, imaging biomarker of coronary inflammation in Europe, UK, and Australia. The European Society of Cardiology has issued very recently a clinical consensus statement focused exactly on how we measure coronary inflammation by using perivascular adipose tissue phenotyping. Now, if this attenuation index predicts future cardiac events, then this has to be demonstrated in prospective studies. And this is what we have done a few years ago with the CRISP-CT study. In this study, we included 4,000 patients, 2,000 from US, 2,000 from Germany, and we have seen that you have this striking prognostic value of the attenuation index on top of age, sex, risk factors, the calcium score, as well as the high-risk plaque, the non-classified plaque. And you can see that you have this improvement of the C statistic by 5.4 to 7.5 units on top of the state-of-the-art of risk stratification you have today that includes risk factors, calcium, and plaque. Of course, when you have a technology like this, you want that to be validated, to be generalizable across the globe, and this is why we launched the Orphan study. That is, it aims for 250,000 patients with coronary CT angiograms linked with prospective outcomes data. We are now at 210,000 patients, and the study is expected to be completed by the end of the year. The first pilot results were published a few months ago from the UK cohort, showing that if you have fat attenuation index score above the 75th centile, you have 10 times higher risk to have a fatal cardiac event if you do not have obstructive disease, and 5 times higher risk if you do have obstructive coronary artery disease. Looking into the specific non-fatal endpoints, you see that if you have high inflammation, if you belong into the top quartile of the distribution of inflammation in any of the 3 coronary arteries, you have between 3.5 and 5.3 times higher risk to have a non-fatal MI. And more importantly, this also predicts ischemic heart failure without the previous formal myocardial infarction. This means that measuring inflammation in the epicardial arteries gives you information about the microcirculation, the coronary microcirculation, that will lead to ischemic heart failure without the formal epicardial artery obstruction. Now, in coronary CT angiography, we have plaque. Do we really need inflammation? Yes, because in the 40% of the patients who have the coronary CT angiogram, there is no detectable plaque at all. And if you look into what has been presented so far, in 8,000 patients from the Orphan study, without any disease, any visible disease, and 0 calcium score, we observed that you have 10 times higher risk to die from a cardiac event over the next decade if you have high inflammation. In the recent paper that was published in The Lancet from the Orphan study, again, if you have small, minor, or no visible plaque, you have 9.5 times higher risk to die from a cardiac event over the next decade if you belong to the highly inflammation group. If you belong to the 40% who has minor plaque, again, if you have inflamed minor plaque, then you have a hazard ratio of 10.4 for a fatal cardiac event. If you do have established disease, then again, this inflammatory metric can stratify you and can position the patients into the correct risk group. And this is done on top of what you can achieve with the high-risk plaque measurements and the calcium score. And indeed, if you add this inflammatory information on top of calcium score, you have a striking improvement of the area under the curve in the ROC analysis. Now, the big question is, how do we treat coronary inflammation? And if we treat them in our trials, for how long? The first results came from biologics. So we have seen some time ago that when you treat patients with psoriasis with anti-TNF-alpha or anti-IL-12, 23, or 17, for a year, you have significant reduction of the fat attenuation index. After a STEMI, you see that within 6 months, you have a striking reduction of inflammation in the coronary arteries. But importantly, not all patients respond well to this optimum medical therapy that is applied after stemming. There is still 20% of the individuals after an ST elevated myocardial infarction who have not responded well to the standard treatments. And these are actually the candidates for novel therapeutics. And these are patients that could be enrolled in clinical trials in the future. Statins, we know very well from a number of published studies, and this is a recently published one, showing that within 1 year of treatment with intensive statin therapy, you have a 30 to 40% reduction of inflammation, which is sustained for at least 3 years. And in a most recent randomized clinical trial that was published a few months ago, just 12 weeks of treatment with ortiguimab, which is an antioxidized LDL monoclonal antibody, has shown a striking reduction of inflammation in all 3 coronary arteries. And this is very important for phase 2 trials where the safety data do not go beyond 12 or 14 weeks. And this is a biomarker that changes dynamically and very fast, and it's an imaging biomarker. With this technology, we are also challenging conventional thinking. We know from the literature that when you expose women with breast cancer to radiotherapy, you increase calcification in the coronary arteries, calcium score. And this was considered to be a sign that radiotherapy causes damage to the coronary arteries. In a recent study that has just came in European Heart Journal last month, we have seen that 2 years after radiotherapy of these women, indeed, you see an increase of calcium score. However, at the same time, you see a striking reduction of inflammation in all 3 coronary arteries, which means that this increase of calcification is actually a sign of stability of the plaques of these women. And when the tumor is removed with the radiotherapy, you have a reduction of inflammation. So actually, the risk should be going down, not up after radiotherapy. Now, inflammation can be combined with the presence of high-risk plaque. And if you have both high-risk plaque and inflammation and increased fat attenuation index score, then your risk is super high. And these are patients who will give events imminently. Of course, smart randomized clinical trials need accurate enrichment tools. And many trials use plaque volume as a tool to identify the right patients. Some others, they use risk factors, risk calculators. Some others, very recently, they use inflammation. But in reality, you want to use all of them. How can you use all of them? It's exactly what we try to achieve with the AI risk model that takes everything into account to find the right patients to be included into our trials. This model that is called CariHeart model, and it's part of a medical device that is used clinically, gets routine coronary CT angiograms, extracts information about plaque, about inflammation, takes into account the risk factors, and give you the absolute risk of the patient for a cardiac event over a certain period of time. The model was trained in the US, validated in Germany, and then it was transferred as a locked medical device for further independent validation in the UK. And indeed, in the orphan study, it gave us a C-index of 0.84 for cardiac mortality. All the guidelines, they tell us about implementing treatments based on the risk cutoffs. And we know that if your risk is low or medium, it's lifestyle and diet. If it's high, it's you start statin or increase the dose of statin. If you are at very high risk, then you consider the additional treatments, and that's where the novel therapeutics will fit in the future, and the biologics that are in the pipeline of development will fit. Now, in the orphan study, this device, the CariHeart device, gives these predicted events in the population over 10 years. These are not true events. These are predicted events. These are predicted graphs. Then we open the labels, and these are the true observed events in the population. So the solid lines are the true events, the dotted lines are the predicted. This tells you that actually, by using this AI technology, you can predict nearly every single event that will happen in this population. And indeed, If we look, for example, into the statin treatment, the prediction from the model was telling us from the trials that have been published recently using the technology that within 1 year you have a 40% reduction of the risk for MACE, which is sustained over 3 years. This 40% reduction is exactly what was observed in the randomized clinical trials with statins a few years ago. So you could essentially predict the outcome of the trials just by following up the patients for a year and recording the reduction of the predicted risk. A similar finding of 40% reduction was observed with the ortiguimab trial very recently, and with the radiotherapy treatment is to 20% reduction of the risk. Now, this technology has been used in a real-world evaluation in the UK's NHS system, where the clinicians were asked to record how this technology changes the management of the patients. And this is the result. It changed the management in 45% of the patients, with 24% starting statin, 13% increasing the dose, and 8% going for additional treatments. This 8% is the target for the novel therapeutics in this population. And before I close, I want to give you a very, very brief overview of what is coming next. Can we become even smarter in selecting the right patients for the right treatments? And I would give you a very, very high-level overview of the concept of radio multiomics. As I have mentioned earlier, depending on the type of inflammation you have in the coronary arteries, you get different changes in the perivascular space. This could be lipolysis, fibrosis, edema, adipogenesis, and angiogenesis. Depending on the type of inflammation coming from the vessel, you get a different combination of these characteristics in the perivascular space. We develop a platform, which is now being tested in a number of clinical trials, that allows us to create customized radiotranscriptomic biomarkers by using RNA sequencing data of the actual arterial tissue as a ground truth to train imaging biomarkers by using radiomics of the perivascular space. Indeed, we have now this library, which is called HbF Heart Vessels and FART library, that combines RNA sequencing data from human vessels with radiomic information. And when you have a new request for a biomarker to tell us whether a certain pathway is active inside the vascular wall, we create a transcriptomic score from the RNA sequencing. We use this as a ground truth, and we then train, using machine learning, a radiotranscriptomic biomarker using as ground truth this information. This radiotranscriptomic biomarker is then tested, validated in the orphan study in our validation toolkit against prospective 10 years or 15 years outcomes. And then we have a validated radiotranscriptomic biomarker to tell us if the pathway that a pharma or a biologic is targeting is active in the vessels of the patients. And then it's being used for early efficacy signals in phase 1 and 2, or to enrich clinical trials in phase 2 particularly. An example of this technology was published in Lancet Digital Health a couple of years ago, showing that you can train a signature that captures specific cytokine inflammation, which reflects the cytokine storm observed during COVID-19. This signature had a striking predictive value, and it could identify during COVID-19 the patients that would respond to dexamethasone. This very same signature of cytokine-driven inflammation is now tested in 71,000 patients in the orphan study from the UK, 5,000 patients from the US, giving striking predictive value in a dose-dependent manner. Just to conclude, Attenuation Index score captures coronary inflammation on routine CT angiograms and is being used in randomized clinical trials, first as a selection or enrichment tool, second as a tool for early efficacy signals, also as an endpoint in phase 2 randomized clinical trials, and in the future, it could potentially be used as a complementary or companion diagnostic for cardiovascular outcome trials. High coronary inflammation predicts cardiovascular events, even in patients with no plaque and calcium score 0, and integrating risk factors, plaque, and inflammation using AI into prognostic models is very powerful to help us understand the events expected in the population recruited into clinical trials and do power calculations. Finally, radiotranscriptomics can potentially transform the way we run the randomized clinical trials in the future. I will leave it here, and we can potentially have discussion.
Thanks so much, Harris, for an excellent presentation. It's such a privilege to hear directly from the person who developed the FAI technology. We appreciate you sharing your knowledge and your vision for where this field is headed. FAI is an example of how imaging is no longer just a diagnostic tool. It's becoming an integral part of how we design and interpret cardiovascular trials. So taking a step back, what is the rationale for incorporating imaging biomarkers in trial design? Firstly, it allows us to directly visualize and quantify biological changes. And with CT, this has the added benefit of being non-invasive. In atherosclerosis, we can use imaging to measure disease progression or regression more precisely via changes in plaque burden, composition, or inflammation, as you've heard. And this often precedes the hard endpoints like myocardial infarction. Having quantitative surrogate endpoints which can offer early insights into treatment efficacy could shorten trial durations and even help support mechanism of action studies. Imaging biomarkers can also help with stratification or enrich a trial for patients with imaging evident high-risk disease. These are the patients most likely to demonstrate a treatment effect, thereby increasing statistical power or reducing the trial size or duration. From a regulatory perspective, imaging biomarkers are increasingly accepted by regulators and beyond endpoints could also serve as powerful enrichment and prognostic tools to help identify and monitor patients. Understanding the science is only one part of the equation. Successfully integrating imaging into trials also requires expertise in trial design and operations. Medpace has been supporting cardiovascular trials with advanced imaging across the multiple modalities: CT, echo, MRI, PET, and invasive coronary angiography. We also have our own embedded cardiovascular imaging core lab. But whether imaging is handled in-house or through a sponsor-preferred external core lab, experience is needed to navigate the imaging vendor relationship, to oversee compliance, ensuring the imaging quality and data flow is maintained, and we have oversight across the multiple partners. From the medical perspective, we have our own in-house imaging certified cardiologists to support trials using any of the cardiovascular imaging modalities. So what do my colleagues and I think about when designing trials with imaging endpoints? First and foremost, we check that the right modality is being used based on the trial objective. It's essential to match the imaging technology to the biological question that the trial is aiming to answer. Each modality has its own strengths and suitability. On a practical level, it needs to be reproducible across multiple sites if we're running a multi-center trial. Does it reflect or predict a meaningful clinical benefit, such as survival or symptom improvement? Is it validated or at least a qualified surrogate marker for a biological process or a traditional clinical outcome? When you image also matters. We plan imaging at baseline and at time points where biological changes are expected based on the intervention. More frequent imaging may give us additional insights, but we have to be mindful that that adds to the cost, the complexity, and burden to patients and sites. In the context of CT, for example, there are ethical implications for repeated imaging due to the radiation exposure and the contrast used. So the risk must be justified by the potential benefit and the scientific necessity. Although you've heard that imaging biomarkers can enrich trial populations and improve signal detection, it can reduce patient diversity into a trial. For example, contrast CT is contraindicated in patients with low kidney function, and sites may not have access to appropriate imaging modalities. Also, when we set narrow inclusion thresholds using imaging biomarkers, that can increase the screen fail rates and potentially slow trial recruitment. We also engage statisticians early to define the variability and the sensitivity of the imaging endpoint, as that can impact the sample size and the power calculations. We consider whether the imaging is used to evaluate treatment responses early and whether imaging thresholds or subgroup based on early imaging responses can be modified using an adaptive design. Now, these sorts of interim analyses and adaptive designs need to be pre-specified and adjusted for multiple looks. So you've heard that imaging endpoints are strategic tools that can sharpen our trial readouts. It enables earlier decisions and can de-risk drug development, but it does require thoughtful planning and collaboration with teams both on the site level, such as having high volume imaging centres, but also having CRO and core lab teams that can help with the operational execution to maintain standardization and quality control. And we do that through adherence to standardized imaging protocols. We minimize missing data, arrange central review of the images, all of which my colleague, Dr. Andrea Flannery, Director in Clinical Trial Management, will discuss now. Over to you, Andrea.
Thank you, Rena. Okay, I will now move on to discuss some operational considerations for clinical trials involving cardiovascular imaging endpoints. First and foremost, it is essential to select the best sites for the success of any clinical trial. Identifying sites to initially contact for feasibility should be data-driven and experience refined to support your feasibility assessment. Data sources may include global health research platforms, clinical study history data, imaging core lab data, central lab data relative to key inclusion-exclusion criteria, and established site network data. For global health research platforms, they can be utilized for public and private health research platforms providing information from millions of investigators, studies, and patients. In addition, very important data is clinical study history metrics. So working with an experienced global full-service CRO provides years of investigator startup, enrollment, retention, and quality performance metrics that can support your data-driven approach to feasibility. Knowing the sites with the correct patient population is also a key factor in site identification. So working with CROs where central laboratories are wholly owned allows for a global repository of lab data relative to the key inclusion-exclusion criteria to drive sound recommendations and target investigators with the appropriate patient population. Another critical data source for studies, including imaging endpoints, is data from imaging core labs. The data from imaging core labs will assess relevant studies, assisting with the identification of investigators in key therapeutic areas with access to specialized equipment, imaging experience, and historical quality metrics such as radiation dosing violations, imaging quality, and image-associated deviations. Partnering with a CRO who have established relationships with individual sites and site networks can also play a key role in being assured sites uphold high standards for startup, patient-centric conduct, data capture, and quality. Having data from these important sources allows for expert team members to then review potential site lists to help create a robust site list for formal feasibility. And we recommend expert team members include at least our medical leads, operational leads, regulatory, imaging, of course, and other key leads within the sponsor, any external vendors, or within the CRO space. And this provides first-hand knowledge of recent site performance and the existing trial landscape to provide input to inform appropriate sites to contact for formal feasibility and qualification. Following the identification of sites, it is critical to start your engagement and training with sites from the very first outreach. This means having a one-team approach from formal qualification of the sites to ongoing training, imaging acquisition, and quality oversight. Looking primarily at the qualification process, it must be confirmed that sites have the capabilities and equipment required for the study protocol. The process for scheduling research patients for imaging assessments and turnaround times for image provision to the imaging core lab also needs to be confirmed as acceptable for the particular study in question. In addition, having at least 2 technicians who have established and good relationships with the PI and study team is paramount. Strong interdepartmental relationships are important to the study with imaging endpoints, as imaging time slots for research studies can be challenging at some sites. Therefore, site interdepartmental collaboration should be assessed and continuously engaged throughout the startup and following activation of a site. If the imaging capabilities are not available on site, it also must be understood how sites will coordinate imaging, including if there is a need for additional contracts, and who will be involved at the offsite facility to support qualification and performance of study imaging assessments. Early mitigation for missing imaging should also be assessed during qualification. The proximity of the site in relation to other potentially selected sites would support backup imaging equipment availability should there be an issue performing imaging at a primary site. In essence, having a one-team approach is important through the life cycle of a study following selection of a site. Here, the training, site scheduling of imaging assessments, and oversight of quality scan acquisition is imperative to protect imaging endpoints in a trial. In addition, retention is a very important factor, particularly if there are sequential imaging requirements in the protocol where both baseline and subsequent imaging is critical to assess imaging endpoints. Okay, we spoke about the important factors to consider for initial site identification and ultimately site selection, and how important it is to lay the foundation of imaging requirements, standards, interdepartmental collaboration, training, and overall quality expectations. This effort starts at qualification and site selection, but needs to be continued through the life cycle of a clinical trial to support continued site training and ensure imaging standardization within the site and across multiple sites involved in the study. During site activation, the clinical trial team, which may include the clinical research associate, the imaging core lab, and or the sponsor, trains and qualifies the sites on the imaging requirements, ensuring the equipment continues to meet protocol requirements, technicians are delegated and trained, and expectations on imaging scheduling are understood and adhered to by the site team. During the site initiation visits, these imaging assessments and requirements should be reinforced to set the site up for success. Once a patient is screened, the monitoring of a site's imaging processes, quality, and scheduling should be continuously assessed to allow early intervention and retraining if a signal of concern is detected. An automated tracking tool should be utilised to support the clinical trial team oversight of sites' overall imaging indicators and whether there are any risks. These can include, but are not limited to, tracking of anticipated imaging, looking at the recruitment and retention data, patient visit scheduling, confirmation of imaging scheduling within the different departments or facilities, timelines for image provision to the imaging core lab, image quality control, and violations of the protocol and imaging manual, which would be captured as protocol deviations. Oversight and tracking of these aspects will allow the clinical trial team to assess where retraining may be required and help standardise processes and quality in a timely manner. Also, the clinical trial team should establish and monitor a set time period from initial training to performing imaging assessments, and the time frame individual assessments are being done. This allows for proactive site retraining to ensure standards are being met and quality is maintained. Finally, and most importantly, is embedding quality risk management at the start of the study and throughout the life cycle of the study. And this is imperative to identify risks associated with imaging endpoints to ensure the well-being of the trial subjects and also the quality of the trial data is maintained. Risks related to imaging endpoints must be identified at the start and continuously assessed throughout the life cycle of the study. Ongoing, centralized and on-site oversight, considering some of the oversight tools I've noted before, should be utilized and utilize technology tools to ensure robust conduct, oversight and reporting of imaging endpoints. Early establishment of triggers for site retraining and intervention is critical to assure standardization and also allow control measures to be implemented proactively and efficiently. Oversight and tracking of triggers through technology tools allows for an integrated method of quality management and promotes oversight activities from all stakeholders involved in the imaging process, such as CRO, imaging vendors, and of course the sponsors. Okay, now I will hand back to Rena for our Q&A session.
Thanks, Andrea, for a fantastic overview and for your operational leadership that's been critical for the trials we've successfully run together. So, as Andrea said, we've got some questions, and the first question we have here is, how do you ensure consistency in imaging acquisition and interpretation across a global trial with multiple centres in multiple different countries? Haris, I think this might be a good one for you.
Yeah, sure. So I think this is a very, very important point that CRO needs to always keep in mind. And when you organize these trials, you need to be very careful in keeping consistency. At the site level, it's important always the scans to be performed with the same equipment, same setting at baseline and at the follow-up. This is partly mitigated, however, at the level of the core lab by the use of robust, regulatory cleared medical devices to analyze the scans, because these devices, they absorb most of the variability that may come up during the trial, coming from acquisition settings, particular local practices, and so on. So it's both the site responsible to ensure consistency, but also the core lab to use the appropriate tools to mitigate for this.
Thank you, Haris. And from an operational perspective, this is absolutely paramount for the study, and again, maintaining that standardization and consistency. So operationally, we would support the imaging core lab in receiving all of those different information. So setting up your systems at the start of the study to integrate the results of the quality control that's being performed, and for ongoing oversight of the QC fails and where there may be warning signs for a clinical site where they may need retraining or early intervention as soon as possible. So we work very collaboratively with imaging core labs to schedule retraining sessions and as well support retraining of the site if we are seeing a deviation from the imaging manual requirements and the protocol requirements. So that is certainly something that's continuously assessed and overseen as early intervention is required to ensure that the site meets the standard that is expected for the clinical trial and it does not continue for future imaging assessments.
Thanks, Andrea and Harris, for those insights. We have time for a final question. If I could ask you, Harris, in a large event-driven cardiovascular outcome study, we're often focused on accelerating event accrual to complete trials more efficiently. Could you elaborate on how imaging tools can be used strategically in trials? You mentioned phase 2, but could it be used in phase 3 to enhance events and reduce trial duration?
Yeah, a well-designed phase 2 trial that uses the appropriate tools to allow you to understand how the intervention changes key features that contribute to events like inflammation or plaque stability, then will allow you to get a very good prediction of the event rates that you expect to see after treating the patients with a certain intervention. So we are doing this in a number of trials now, where we use the AI risk model as an endpoint in the phase 2 trial. By doing that, you get the effect size on the risk. And then you say that the 5-year risk or the 3 years risk goes from 3% to 1.5%. So this means that in 3 years' time, you will save 50%. You have a reduction of the events by 50% or something like that. And then we are doing this in a number of trials, but of course we haven't translated it directly into, we haven't validated directly into cardiovascular outcomes trials for now. The only validation we have is by estimating the risk reduction for interventions where we know the results of the outcomes trials, and we see that they align very well.
Okay, well, that's thank you very much, Harris. So that wraps up our question and answer session. Thank you very much for listening, and I'll hand it back to Sonia to wrap up.
Well, thank you very much for answering all those questions. We appreciate that. We've reached the end of the Q&A portion of the webinar, and if we couldn't attend to your questions, and there's a number of them out there, rest assured the team at Medpace will follow up with you after this presentation. Now, if you have any further questions, direct them to the email address you see there on your screen, and that's info@medpace.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our webinars. Additionally, I'm going to send it to you right now. You should see it there in your inbox, a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until the next time, please take care and bye for now. Bye, everyone.